Hey, this is George Edwards here with another research update. I've repeated my last experiment where I showed a strong ERK phosphorylation in response to a protein S ligand treatment in MER knockout bone marrow dendritic cells, which was interesting because it showed there could still be cell signaling in an acquired resistance pathway through the TYRO3 receptor in the same family. So I have a Western blot that's currently ready to image, so let's go see if the data repeats.
She could be anybody, then she'll be the only
I wanted to take a moment and take you through the broader pathway that I'm doing cell signaling Western blotting analysis on. So, pretending that this is a bone marrow dendritic cell, we would have MER, AXL, or TYRO3 TAM receptor tyrosine kinases in the cellular membrane, and they can signal through the RAS and RAF proteins, and also phosphorylate the ERK protein that I'm currently studying in my experiment now. And we can also see downstream metabolic pathways that can be affected through ERK signaling. So, looking at this other figure here, we can see that the ERK protein can actually stimulate fatty acid oxidation, which is something that I showed as an effect through dead cells in my chalk talk, where I showed that dead cells were increasing the amount of mitochondrial activity through a seahorse metabolic assay. So essentially what I'm trying to do is trace this pathway in reverse all the way from its functional effects all the way to where it starts with the root of the receptor tyrosine kinase and being activated through either oxysterols or ligands such as protein S or Gas6. So you might be wondering, well, what does all this actually mean for cancer patients? Well, for patients who are receiving anti-PD-1 immunotherapy, the goal is to effectively restore their T cell function. For T cells to function, antigen-presenting cells need to function well too, so that they can present antigen to those cells. For dendritic cells, a class of antigen-presenting cell, they need a glycolytic surge in their metabolism to effectively present antigen to T cells. I've shown that by treating dead cells in a co-culture with dendritic cells, you can actually have a spike in mitochondrial respiration, and I've also shown that dead cells can increase the amount of ERK phosphorylation, which can feed into metabolic pathways. Recently, I'm trying to show whether differential expression of the TAM receptors can affect this same dead cell-induced ERK phosphorylation. So ultimately, the goal of my experiment in the long term is to give some substance to different small molecule inhibitors that can hit different TAM receptors differentially as a way to potentially counter acquired resistance. So I finished imaging the repeated Western blot, and this is really the moment that I look forward to. All the hard work culminating in a research result that we can analyze. So when we're looking at repeated data, obviously we're looking for comparisons, and it's to be expected that the results will not be exactly the same, but I think it's important to realize the amount of variability that can add up over the course of such a long experiment and so many steps. This can not only occur from the actual cells themselves and factors such as the lysis buffer, but also the actual transfer of the Western blot from the gel to the membrane could affect protein levels. So looking at the data, I've set it up very similar to how I set up my previous slide. The main difference is that I included another image here of an exposure that was 30 minutes for the blot that had the MER and TYRO3 double knockout cells. So immediately I kind of went to this ninth lane where I pointed out there was an interesting finding having phosphorylation of the MER knockout BMDCs with a protein S treatment. And again, I do see staining compared to baseline, which I wasn't seeing much baseline phosphorylation again in these blots. And another interesting lane is down here, where you can see that in the MER knockout cells, we're also getting some staining with the Gas6 ligand. The Gas6 ligand has more of an affinity for the axil receptor, so the result of this experiment is kind of telling me that maybe signaling that causes a metabolic switch and unfavorable antigen presentation in dendritic cells could involve the axil receptor as well as the tyro3 receptor. And we've had some data that's come up in recent lab meetings that talks about potential negative effects of the axil receptor. So this could be another piece of substance to support that. Something else that I see in the data is generally having a stronger band when we have ligand and the dead cells in the MER knockouts. Generally, I'm seeing less of a strong effect from the dead cells on ERK phosphorylation in the double knockout cells. So we'll need to investigate that a little bit more. But overall, I'm very pleased with how the experiment went. Probably with an experiment like this, have to repeat it more than just once or even twice. And we'll have to go to the drawing board this week and talk with everybody in the group and get their thoughts. But overall, it's been a great past 2 experiments. I recently spoke with Dr. Lloyd Hay, a scoliosis surgeon in the Raleigh area who founded the Hay Clinic for Scoliosis and Spine Care and the CareGuard Institute, which is an organization for pre-meds and medical students to promote quality improvement in the medical field and compassionate care. I asked him how I could stay involved with the program once I start medical school. He told me that if I were accepted to the UAB MSTP, he'd be thrilled if I could start a CareGuard Institute chapter at the school for the pre-meds and med students. Let me take you through the website and show you some of the things we could do in such a chapter. An integral part of the CareGuard Institute is reviewing lectures by Dr. Hay and other guest speakers to learn about how they maintain a strong seed of compassion and avoid burnout in their careers. Dr. Hay keeps his faith and engineering mindset close to heart as a way for him to recognize the fragile and complex nature of his precious patients, and I plan to use these strategies as well to avoid burnout in my career. Medical students in pre-meds can get Level 2 and Level 1 certifications respectively through the CareGuard Institute on the courses provided. Part of how our nonprofit inspires members to affect change are through quality improvement projects and educational interventions, such as how would a shared decision-making infographic help patients decide whether they should pursue surgery versus conservative therapies such as bracing or short physical therapy? Or how can we help pediatric clinics promote early screening during Scoliosis Awareness Month in June? Part of our mission is to promote the use of standard work to eliminate unsafe variability in medical treatment. Dr. Hay, as a pilot, uses aviation-inspired safety checklists in every surgery to ensure the highest level of care for his patients. Part of our clinical research goals are to validate the use of these checklists and convince other healthcare practitioners to use similar versions of standard work no matter their specialty. I'd like to close by telling you a little bit about my relationship with Eric Ubel at UAB. While Eric was never actually my research advisor when he was still at UNC, he always felt like a colleague and close mentor to me, and we would talk not only about science then and now, but also just about life in general and about our similar interests. For example, I know that Eric really enjoys doing Ironman competitions and triathlons, and we always had a good time talking, and I feel like he would be an amazing mentor to me if I were at UAB and doing a research rotation over the summer. He just had to make noise. Part of how our nonprofit inspires members to affect change are through quality improvement interventions and quality improvement projects. Leave it. Part of how our nonprofit inspires members to affect change are through part of how our you gotta leave, man. You gotta leave.